Hello, I'm Dr. Edwin George, and today I'm going to talk about biomarkers and what they can tell us about multiple system atrophy. Uh, Disclaimer, I work for the FDA these days, but this is talk about basic science and not about policies, and nothing I say should be considered to represent the FDA's views or policies, and I have nothing to disclose. So biomarkers are mentioned everywhere in the medical literature and in press releases about multiple system atrophy and other diseases these days. But what are they? Well, bio for biological, and markers. They mark something as being present or absent, or they mark a measure of something. Lots of things can be used as biomarkers. So here's a formal definition of a biomarker. This is taken from a booklet that's published by the NIH and FDA working group, BEST, Biomarkers, Endpoints, and Other Tools. And they say a defined characteristic that is measured as an indicator of normal biological processes or pathogenic processes or responses to an exposure or intervention, including therapeutic interventions. And they point out that there are multiple types. They can be molecular, they can be histologic, which means that you can see them under a microscope. Radiographic, you can see them in X-rays and MRIs and other imaging. Or physiologic, actual measures of your biologic function. And biomarkers can measure disease presence or status. So whether it's there or not, but also how it's progressing is the status. And they can measure aspects of response to treatments. A biomarker is not an assessment of how a patient feels, functions, or survives, which is an important thing to remember. Biomarkers are very useful in clinical studies of how diseases progress, whether therapeutic interventions work, and so forth. But the ultimate test of a treatment is that it makes you feel better, function better, or survive better. And the fact that some chemical in your blood has improved as response to the treatment, if it doesn't ultimately lead to how you have an improvement in how you feel, function, or survive, it's not a treatment. So biomarkers are useful tools in looking for treatments, among other things, just understanding diseases. But they're not the end point of trying to demonstrate that something is a treatment. Now, there are lots of types of biomarkers, and this says biomarkers classifications, but you'll see over on the left side of the screen, over the statement, things to be detected or measured, we have gene-based, and you can measure nuclear DNA, you can measure mitochondrial DNA, you can measure RNA, you can measure microRNA, different types of genetic material. They can be protein-based, they can be regular proteins, they can be peptides, small protein fragments, many of which are signaling peptides, so they become important as biomarkers. They can be antibodies as a measure of your immune response. They can be metabolism-based, lipids, fats and cholesterol, carbohydrates, sugars. They can be enzymes, things that proteins that your body uses to make certain processes occur. They can be metabolites, breakdown products from things you've eaten, from drugs you've taken, etc. So those are all things to be detected. And then we get the yellow circle near the middle that says source base. And underneath that, I've said where. This is where you're measuring these things. All those things over to the left are being measured, but they can be measured in blood, they can be measured in serum, they can be measured in plasma, they can actually be measured in tissues, and they can be measured in cerebral spinal fluid. So they're not independent categories. Usually you need some combination of the three to the left and one from the yellow to have a biomarker. And disease-based is why you're looking at them. And so once you've defined that you're looking at a protein in blood, why are you looking? Well, you can have prediction biomarkers. You can have detection biomarkers. Detection biomarkers are things you can use for diagnosis. So you have diagnosis biomarkers, and you have prognostic biomarkers, things that you measure that might tell you what's going to happen, particularly with the disease process. And so there's lots of different reasons related to diseases why we want to look at biomarkers. And then sort of other categories over there to the right, imaging biomarkers. Everything on the far left is a chemical that you can measure somewhere in the blood or the tissue or the spinal fluid, whatever. But actually on images from MRIs or from X-rays, you can see things that count as biomarkers. In silico biomarkers is a cute one. In silico is a popular term these days for something that happens in a silicon chip, so a computer-based biomarkers. So we're really talking there about an index that you might take measurements of a whole bunch of things and weight them differently and add them up and come up with something, an index that you say is a biomarker. Or you could have a pathological biomarker where you actually look under the microscope. And this is the cytological that was mentioned earlier, or histochemical histology, things that you see actual tissue under the microscope where you might be looking to detect certain things are present by using special stains. And in fact, we do this a lot in multiple system atrophy and other Parkinsonian diseases with the skin biopsy tests, where we're actually looking to see the abnormal alpha-synuclein in the skin, but we're not just looking to see if it's there or not. We can also see where it is, if it's near blood vessels, if it's right under the surface of the skin, to name a few places, which says something about which kind of nerve tissues it's showing up in and tells you more than just knowing that it's there. So the skin biopsy is a kind of biomarker as well. And here's a whole bunch of examples from around the whole field of medicine and biology. So you can have a susceptibility biomarker. There are certain genes that tell you that you have a strong risk of developing a particular disease, such as ApoE4 can tell you whether you're likely to end up with Alzheimer's disease. You can use biomarkers for diagnosis. If you have a particular gene, the SMN1 gene, then you have spinal muscular atrophy, or at least if you have the right mutation in it. Prognosis, you can predict, will you live longer? And in Huntington's disease, we have triplet repeats in the genome, and the more repeats you have strung together, the more severe the disease was going to be, and the shorter period of time that you will probably survive with the disease. We can have prediction biomarkers that I mentioned earlier, and these are often used to measure response to treatment, which can be good or bad. That is, we can look, again, typically at certain gene features and say, well, this treatment won't work for you because of the genetics that you have. An example of this is some of the blood thinners that we have just don't work in certain people because of their genetics. Or it can predict that this treatment is likely to cause a side effect. So both can be possible. If we continue around clockwise, we get PD/response. PD here, unlike most of the times you'll see it in this talk, stands for pharmacodynamics. Pharmacodynamics is basically the measurement of responses in response to treatments. So response, and tells you whether or not a treatment worked. And there's an example here that a particular substance in the serum tells you whether or not you're likely to get amyloidosis. And if you do a treatment to try and affect it, you can change that measurement in the serum. Monitoring status of the disease, as I mentioned earlier, is really the progression of the disease. Or you can measure something that changes with drug exposure. And the example given here is on MRI. You can very easily follow multiple sclerosis, is what MS stands for here. You can see different numbers of lesions and their size in the brain and the spinal cord and so forth. Safety. They can actually tell you if you're having an adverse event. So hepatotoxicity means liver toxicity. Lots of drugs have the potential to cause liver toxicity under certain circumstances or in certain people. ALT is alanine aminotransferase. It's one of the liver enzymes. And we typically look for some of these liver enzymes in your serum to find out if your liver is healthy, because if it's not healthy, it will tend to leak the enzymes. And that can be an adverse event if you're taking a medication that might cause liver disease. You could detect it before it causes any symptoms, which is a very useful use for a biomarker. And often there's a lot of medications that when people start taking them, will measure your liver enzymes every so often for at least the first several months to see whether you're someone that this drug is going to cause a problem with your liver. So lots of useful biomarkers already out there. But we want to talk about multiple system atrophy. And one place where you may have come across it is if you've looked at the Movement Disorder Society criteria for diagnosis of multiple system atrophy. The criteria include all sorts of things to try and determine who has multiple system atrophy versus some other Parkinsonian or cerebellar type of disease. But one of the things that are amongst the classification criteria for deciding whether people have particularly what's called clinically established multiple system atrophy. So you have a number of features, and then you add in MRI markers to try and distinguish that, yes, this is truly multiple system atrophy or it's not. And as many people know, and probably many of you have experienced, pinning down the diagnosis of multiple system atrophy is often quite difficult. So for MSAP, Parkinsonian MSA, on the left side, we have atrophy of various structures in the brain. We have something called the hot cross bun sign, and we have changes in the diffusivity in certain areas of the brain. On the right side, we have for MSAC, the cerebellar type of MSA, atrophy of some of the same areas, but also some other areas. We can still get the hot cross bun sign with either type of MSA, but it's considered to be typical for MSA in general. And we see changes in diffusivity in some structures of the brain as well. And these are little pictures that are taken from the description of the criteria to show you what some of these things are about. So if you look up in the top left, there's a box that's labeled 1, and it has an MRI image of an MSA brain and a Parkinson's brain, and you want to try and tell them apart. Well, on the MSA image, there's an orange arrow coming in from your left, pointing at the pons, which is this oval-shaped structure. And there's another dotted arrow coming up from the right, which is pointing at the what's the fourth ventricle, which is a triangular dark space right there, which is a little hard to make out because the head of the arrow is right on top of it. But you can see that the whole space is dark, and that's a fluid-filled space. So if you compare to the Parkinson's disease, the pons is nice and fat on the Parkinson's compared to what's there on the multiple system atrophy patient. And this space that's the fourth ventricle is, actually I go this way, you can see it again over on the Parkinson's side, and it's a much smaller dimple because right behind it is the cerebellum, and the cerebellum has atrophied in the patient with MSA. And the final dashed arrow coming from the right, but downwards, is pointing at what are called the folia, the folds in the cerebellum. And you can see that they're kind of thinner with more dark space between them because the cerebellum is atrophied. And when you look at the Parkinson's image, the cerebellum is nice and plump with just thin lines of darkness between the folia. And you can see this even better in box 2, which is the top center. Didn't mean to go to that yet. So again, we have a section through MSA brain and a section through Parkinson's brain, looking closely at the cerebellum as it comes off of the brainstem. And the solid arrow is pointing at where it comes off the brainstem. You're looking at some white matter, which stretches out as a band with the folia poking up off of it. And you can see the solid arrow is pointing to the same place in the Parkinson's, where it's a very thick structure as opposed to the atrophied thin structure in the MSA. And again, you can see the dotted arrow on the MSA patient is showing how atrophied the folia are. These little fingers sticking up with lots of dark space between them, as opposed to the fat fingers on the Parkinson's disease image with very little space between them. And then number 3, top right box, is showing us the hot cross bun sign, which there's a kind of pale cross on this section through the brain stem that's actually hard to see. The arrow is pointing in the general area, but we can draw where the cross is, and then in the very center of the screen is a blown up version of the image, and you should be able to see some of that white cross appearing where the orange arrow is pointing. On the bottom left, we have 2 images of multiple system atrophy and 1 image of Parkinson's, and we're looking at diffusion images to look at diffusivity. And if you look closely where the orange arrows are pointing on both the MSA brains, you'll see some dark and bright adjacent bright areas reflecting increased diffusivity. And you don't see that with the orange arrows pointing in roughly the same place on the Parkinson's brain. And finally, number 7, bottom right, is again 2 multiple system atrophy brains and 1 Parkinson's brain on an MRI sequence that's meant to show iron. And what's easiest to see here, you can see on both of the MSA images, there are upper and lower arrows, upper solid and lower dotted, and on both sides you see dark bands extending between the upper and lower arrowheads on each side of the brain. And this reflects deposition of iron in the area of the brain. And you can see there are no dark bands on the Parkinson's. So lots of different things that you can see that have been picked out as biomarkers for making the diagnosis of multiple system atrophy from MRI scans. So imaging can be a diagnostic biomarker, but can it be a disease progression biomarker? Well, things that are atrophying tend to atrophy more over time, so you would think they might. And this is from a very recent paper in trying to demonstrate a change in the actual volume. So this is volumetric MRI. And you can see the top left, they're measuring total gray matter. And gray matter is any part of the brain that's mostly made up of cells, as opposed to white matter, which is mostly where the connections run between different parts of the brain, like bundles of wires. So when you're looking at total gray matter, you're looking at loss of cells throughout the brain. And you see we've got two sets of boxes, one that's at 3 months, it says, or at 6 months, it says month 6 and month 12. And the gray box are healthy controls. And they actually measured them obviously at month 0 as well as 6 and 12. But what's being shown here is the percent change in volume. So the gray box at 6 months is right at the 0 mark on the vertical axis. It hasn't changed from 0 months to 6 months, whereas the other 3 boxes have shown some reduction in volume. And the 3 blues, the darkest blue that comes first is the navy blue going left to right. That's all of the multiple system atrophy patients they were studying. The what I'll call royal blue in the middle is the patients who have MSA-C, and the sky blue or pale blue on the right is patients who have MSA-P. And you can see all of them have decreased at 6 months, and all of them have decreased even further at 12 months. So you do see a change over time in the volume that you can measure in these parts of the brain. Even the healthy controls, they're age matched, and so they're an older group of patients, and they have a little bit of loss of gray matter by 12 months. If we look on the top right, they're looking at striatum. The striatum is a very crucial place for the connections of dopaminergic neurons and other neurons involved in Parkinson's and Parkinsonism. And so again, the healthy controls don't change much, but and at 6 months, we see that there's some reduction in volume in all of the multiple system atrophy patients, perhaps a bit more in the MSAP patients. But you can see definitely when you get to the 12 months, the MSA patients in general are lower, and it's mostly being driven by the MSAP patients in the sky blue, and the MSA cerebellum are getting left behind. So this is a good area to look at to see progression of MSAP, but not necessarily MSAC. If we look in the bottom left, we see brainstem volume. And again, this is near the ponds where we were looking before. And you can see again a general decrease in the MSA patients. And this one, when you get out to 12 months, it's clear that the MSACs are pulling ahead compared to the group as a whole in the navy blue, and the MSAPs in the light blue are lagging behind. And so a better place to look if you're trying to track progression of MSAC. And then in the bottom right, they're looking at gray matter again, but specifically in the cerebellum. And as you might expect, again, everything's progressing a bit at 6 months, and by 12 months, MSAC is now pulling well ahead and driving most of the change compared to MSAP. So you can track the progression of disease. They actually looked at many more areas than this, but these are some key areas I pulled out to show you their graphs. Another type of image you can use is a DAT scan, which probably many of you have had. And so this is looking at the percentage change in DAT scan specific binding ratios in the striatum again. And this, again, it shows the change in all types of multiple system atrophy, not so much in the gray box representing the normals. And particularly, you can tell at 12 months, the change of the whole group in the navy blue box is driven primarily by the very pale blue MSAP, which you would expect because this is where Parkinson's symptoms tend to arise from in the brain. So another type of imaging that can potentially be used to track progression. So imaging can be a diagnostic biomarker, but it can also be a disease progression biomarker. If you have enough people, that was 41 MSA patients. I think 26 were MSAP and 15 were MSAC. But they did some calculations, and if you want to really be able to tell a difference in the average of how a group of patients are progressing, for most areas of the brain, you're going to want 100 to 200 patients versus the 15 to 25 that we were looking there. You could also use better imaging. These images were all done with the standard 3T magnet. The magnet in the MRI machine, the stronger it is, the sharper the images and the more detail you can see. And there are stronger magnets, but the 3T most places have, so you can have lots of research sites to follow a bunch of patients. If you want to use the stronger magnet, you might have to start flying your patients to a center that has a stronger magnet. And finally, we want to look at things that are in blood and spinal fluid. And so I picked two representative. There's a whole slew of things that have been looked at as biomarkers in MSA and in other neurodegenerative diseases. Neurofilament light chain is a major component of the filaments, neurofilaments, that provide the structure in nerve cells. And so it's released when nerve cells break down or degenerate. And neurofilament light in particular is more soluble, and you can measure it in spinal fluid and in blood. Now, all neurodegenerative diseases, by definition, have breakdown of nerve cells. So neurofilament light is a major biomarker, but it's not necessarily terribly specific for one disease or another, but it behaves differently in different neurodegenerative diseases. Monocyte chemoattractant protein 1, MCP1, also known as CCL2, plays an important role in inflammation in many parts of the body, including brain. And recently, there's been some research about the importance of this biomarker in the MSA, but remind you that there are many different biochemicals that are involved in inflammation in the brain. And so there's a whole slew that have been measured. This is just as a representative sample. So one thing, this is a group, they actually looked at 6 different biomarkers to do their paper, but pulling out 2 of their plots, you can see that MSA, which is right in the middle, has lots of NFL on the left and lots of MCP1 on the right, compared to a whole variety of different neurodegenerative diseases, including things like progressive supranuclear palsy and Parkinson's disease and also Alzheimer's disease. So the idea is that with 6 of these, you might find certain patterns that you could say, this is what the diagnosis is. However, going back and looking at NFL in MSA patients over time, same 6-month and 12-month plots, you see that we don't see the normal controls here, but these are all higher than normal, as we could tell if you look on the left plot over here. It's higher than normal. Farthest to the left is normal. So MSA generally has a lot of neurofilament light out in the serum or in the blood, and also in the CSF, but it really doesn't seem to change much over time. Maybe a little. But what you do find, so this is splitting up the amount of neurofilament light measured in different multiple system atrophy patients. And the blue line shows patients who have less than the median. So the median is not the mean. It's if you line up all the values you've got and pick the one that's closest to the center, that's the median value. And so these blue ones have levels of NFL that are lower than most MSA patients. And the red ones have NFL values that are higher, in the higher half of the patients. And you can see that if you have less NFL, then your disease progresses more slowly and deaths are less frequent than if you have lots of NFL. So we can say now that NFL and MCP can be diagnostic biomarkers to some extent, but NFL is consistently elevated in MSA, might not change much with disease progression. MCP is one of many proteins involved in the inflammation that we know is active in multiple system atrophy, but its exact role in relation to disease progression is still under study. Some series suggest that maybe it's more in MSAC. Some people think it's more in early disease rather than later. But NFL can be a prognostic biomarker predicting how disease will progress. I want to mention this is not something that's generally done clinically, but it's something that is useful to know, particularly in research, because if you're going to do a study, you might want to have people who are likely to progress faster so you can pick up changes faster. Or you might want people who progress slower because it might be harder to slow down with a treatment a more severe disease. That needs to be sorted out. The final important thing to know about biomarkers, here's an example of one model of how inflammation works in MSA. You see the white with the green nucleus nerve cell with its long connecting axon swinging off to the right. You see the small, dark, spiky, healthy microglial cell, and you see the green, healthy oligodendrocyte that sends processes over and makes the myelin wrappings on the axon from the nerve cell. And then in panel B, you see lots of alpha-synuclein oligomers, which both get into and are released from the affected oligodendrocytes. And in the oligodendrocytes, they can clump and make what are called glial cell inclusions, which are a pathologic hallmark of multiple system atrophy. But they can also activate the microglia, and the activated microglia release pro-inflammatory cytokines and reactive oxygen species. And the idea is that these cause promote the inflammation that damages both the myelin and the axon connection from the nerve. And so there are questions as to really where the inflammation acts in the disease mechanism. So you can see going right across the midline of the slide, we have various triggers causing alpha-synuclein aggregation, causing cellular dysfunction, and eventually neuronal degeneration. And you have a treatment that's supposed to affect inflammation. If inflammation is near the beginning, sort of the way the previous diagram was suggesting, then we first of all expect it to affect inflammatory biomarkers such as MCP1. So we'd expect it to be perhaps lowered. We often talk about this as target engagement. It's just measuring that the treatment actually has an effect on inflammation. But we also expect that symptoms will perhaps be better and NFL may be lower because they're downstream of the damage that the inflammation is doing. So if you can interfere with that process, there'll be less neuronal degeneration, less progression of symptoms, and less NFL released. Alternatively, other people have suggested that inflammation comes later in disease and that only once the nerve cells are really dysfunctional is the inflammation triggered, and then that leads to the degeneration of the nerve cells. But little trick here, if the nerve cells are sufficiently dysfunctional, that may give you all the symptoms, whether they've broken down yet or not. So here you still see the target engagement for treatment one affecting inflammation. You still see NFL, downstream effect of neuronal degeneration, but perhaps in this model, the treatment would have no effect whatever on the progression of symptoms. We could do another one with a different treatment that's supposed to act directly on the cellular dysfunction. This is where many things that you hear referred to as neuroprotective would be expected to work. And so then it's going to affect everything, even though inflammation is later. So the biomarker will be affected if the inflammation happens because of the damage to the nerve cells. The neuronal degeneration will be slowed down or won't take place, so the NFL might go down. And symptoms might be better if they're driven by the cellular dysfunction. And finally, a third treatment situation where the inflammation starts early, but mostly leads to neuronal degeneration. The treatment is focused on preventing neuronal degeneration, so it affects the NFL biomarker, and the inflammation is still there, but would be unchanged, and so the MCP1 would be unchanged, but the symptoms wouldn't be changed at all because they're coming from the cellular dysfunction, which is upstream of the complete degeneration. So this is very useful because it helps us to sort of pull apart what actually happens in the mechanism of disease and multiple system atrophy. We often try and figure those things out in animal models, but disease in animals is often not quite the same as in actual human beings. But when somebody finds a treatment that affects a biomarker, you often get an announcement that makes it sound like, oh, this is going to be a great treatment and it's going to give clinical benefit. And it's not necessarily so. Sometimes it does, and sometimes it doesn't. So it is exciting. And when we do find something that really leads to a good beneficial treatment, we'll probably see it affecting biomarkers. But when it doesn't work out that way, but the biomarkers are affected, it pretty much always advances our understanding of the disease, and that brings us closer to a treatment with clinical benefit. So it's still exciting and a great thing. So I hope I've clarified what biomarkers are all about, what they're good for, and how much you can depend on them to tell you one thing or another about various treatments. Thank you for your attention.